So keeping this in mind, I have chosen a very important and pertinent topic for today's presentation. And let me share my presentation and these through slides. Let me start my presentation so that you will be able to understand the concept what I am going to share with this afternoon. So again, I welcome each and every one of you. Hope you are able to see my presentation.
Yes, sir. It is visible, sir, and your voice is also audible, sir. You can go ahead, sir. Thank you, sir.
Thank you very much. Thank you very much, Dr. Basurajappa, once again. Okay, and 1 second, I will just set few things.
So you are
So as you know, since independence, Indian agriculture, succeeded in achieving the green revolution. Credit goes to the eminent scientists, the farmers, and so many other stakeholders to achieve green revolution in India. Because of that, India today is self-sufficient in terms of food production. Agriculture is an important sector in the Indian economy, and about 42% of the rural populace still depends on agriculture. India is the world's largest producer of many fresh fruits, and we are number one in production of major staple foods at the moment. Maybe years to come, India will be number one in terms of agricultural output. The total food grain production in the countries is estimated around 314 million tons, and 90% of our food comes from about 10 to 15 crops. Among these 10 to 15 croplands, potato is one of the important vegetable food crop, botanically known as Solanum tuberosum. It is one of the staple food crop all over the world. If you look at its history, potato is a native of South America. It was first domesticated in Peru. There are about 5,000 varieties of potato worldwide. Actually, Spanish introduced this potato crop to Europe and in the second half of the 16th century. And because of similar weather conditions favorable to potato growth and development, and it became number one crop throughout Europe, and it is also one of the number one crop in rest of the world. And today, the China is the number one country in terms of production. India stands second in terms of production. It was in 1842, the potato adjusted to European conditions so well, and the people of Europe, particularly Irish people, they were all dependent on this important crop. It was during 1842 to 45, a late blight disease, which has devastated the vast areas of potato crop in Europe. The fields which promised bumper harvest, transformed into a rotten field within a span of 15 days. All tubers inside the soil were spoiled and started emanating a very bad smell. When the farmers, the Irish people, when they digged out, they could able to see only the spoiled potato tubers. And some of them, because it was the only staple food, some of the farmers do eat the spoiled ones. And soon the hunger, dysentery, and typhoid spread among the farmers of Ireland. And 1.5 million Irish people died due to hunger and malnutrition, and the same number of people migrated to other parts of the world. This disease in the history, if you just read, it is better known as Irish famine, and it has destroyed the potato cultivation in Europe. The fields which are promised bumper harvest transformed into a rotten fields. British royals who brought an eviction act during the same time, and the tenants were thrown out of their homes in Ireland for not paying the rent. Finally, the help came from the United States of America and also Canada, where 1.5 million people left home land, Ireland. The Ireland population, which was once upon a time, it was 14 million, reduced to 7 million. That speaks about the severity of the late blight disease, Irish famine. It is the Irish famine which completely devastated the social fabric and economy of the country, Irish people. The tenants were thrown out of their homes in Ireland for not paying the rent, and it was also published in Illustrated London News. And the homeless families had to find a shelter in the countryside. Nearly 1.5 million people died, and the same number of people migrated to other part of the world. Since appearance of late blight disease in 1845 to 49, it took another 15 years. It was in the years 1861, Anton Debary, a German botanist, he did a simple experiments with a late blight disease of potato, and he was the first person who isolated the fungus responsible for the late blight of potato disease, and he named the causative agent as Phytophthora, meaning plant destroyer, and responsible for the disease. And he laid the strong foundation for the development of plant pathology, systematic study of plant diseases, and that is the starting point for the origin of plant pathology. In the botany and also in agriculture, plant pathology is one of the important disciplines which take care of plant diseases and how to manage plant diseases caused by different causative agents. Plant pathology or phytopathology, it is a science that studies plant diseases, and it tries to attempts to improve the chances of survival of the crop plants. It has a practical and noble goal of protecting the food available for humans and animals. Therefore, it is a very interesting, very challenging, and worth studying subject both in botany and also agriculture. The scientists or the professors who study plant diseases are known as plant pathologists or the plant doctors. The task and aim of plant doctor is to keep the plants healthy so that we always get healthy food, and it very important for healthy society. So the plant pathology calls for knowledge of pathogens that cause diseases in plants and how do they affect the plant growth, yield, and quality of the harvested product. There are different types of causative agents responsible for different types of diseases in crop plants. All these can be classified into two types: non-parasitic diseases and parasitic diseases. Non-parasitic diseases, otherwise known as abiotic disease, caused by excess or deficiency of minerals, excess of excess of toxic chemicals, chemical pollutant, extreme environmental condition, air pollutant, excess pesticide, fertilizers. So all this comes under non-parasitic diseases. We have parasitic diseases always caused by living organisms, them as biotic agent and also mesobiotic agents. These includes the fungi, bacteria, algae, protozoa, angiospermic parasite, insects, phytoplasmas, and viruses and virides. Though they comes under mesobiotic group, I have classified all of them under parasitic diseases because all these need a living host for completion of their life cycle. So let me not dwell upon all those things. I'm interested in only the last important thing, viruses and virides. Let me introduce briefly what are viruses, and I will take a lead from that viruses, and I will introduce my topic to you, why virides are otherwise known as infectious RNAs, why they are important in present day context. Viruses are nothing but nucleocapsid particles. They are made up of nucleic acid, either DNA or RNA, externally surrounded by a protein coat. The nucleic acid sequences that ranges from 1000 to 250,000 base pairs, starting from 1000 to 250,000 base pairs or even more. They are ultramicroscopic That means you need a electron microscopy to visualize their presence in the cells, plant cells or animal cells. They are acellular in nature. There is no cytoplasm or protoplasm in them. That's why they are also considered as acellular in nature. They are unable to grow on liquid, medium, inanimate media. Therefore, they are non-culturable and on inanimate media, and they are obligate intracellular parasite. They need a living host for completion of their life cycle. It was Adolf Meyer, a Dutch botanist in 1886. He first observed the tobacco mosaic disease. That was the first viral disease recognized by Adolf Meyer. It was in Dmitry Ivanovsky, a Russian botanist, 1892. Isolated the sap. He said that the sap of the tobacco mosaic plant is infectious and able to cause disease in healthy plants. It took another 20 years. Okay. It was W. Bejernik in 1898 called this infectious material Contagium vivum fluidum. That means living infectious fluid. The causative, the sap which is able to cause disease, he called that living infectious fluid as virus. The name virus derived from Latin word means living infectious fluid responsible for causing disease. W.M. Stanley, it was in 1936, first person purified the tobacco mosaic virus. That is the starting point for the development of virology. So all viruses that we know at present, they were discovered after 1936. The first to be crystallized in the form of a protein, it was only in 1936. Viruses can be classified into basically three types. Animal viruses, in that DNA is normally the genetic material. Certain exceptions are there, like retroviruses, though they are animal viruses, like HIV, the genetic material is RNA. And also SARS-CoV-2, okay, it is also a animal virus, but genetic material is RNA. Certain exceptions are there. Then we have a plant viruses. In plant viruses, always the genetic material is RNA, except certain virus, exceptions are there. Gemini viruses, wherein DNA is the genetic material. Then we have bacteria infecting viruses called bacteriophages, where DNA is the genetic material, and there are certain exceptions. But based on this classification, we can say that animal viruses are generally made up of DNA as genetic material, plant viruses, RNA as the genetic material, and bacteriophages, DNA as the genetic material. Look at this animal virus influenza. In the central place, you have nucleic acid surrounded by a protein coat. In turn, it is also surrounded by an envelope, which is made up of lipoprotein in nature. Look at this HIV virus, centrally placed nucleic acid, RNA, okay, surrounded by a protein coat and externally surrounded by a lipid membrane of human origin. Look at the plant virus. Plant virus, tobacco mosaic virus, it is a rod-shaped virus. Look at the centrally placed RNA, externally surrounded by a protein coat. So that is why all viruses, whether animal viruses or plant viruses or bacteriophages, they have a centrally placed nucleic acid externally surrounded by a protein coat. That's why they are referred to as nucleocapsid particles. No one ever imagined that nucleic acid, specifically RNA, alone could be able to cause diseases in plant. We all believed that both nucleic acid and surrounded by a protein coat, both are very essential for survival of this particle and able to cause diseases. Only in the 20th century, later part of the 20th century, the experiments proved beyond doubt that even a small piece of RNA, smaller than the viral RNA, that cause plant diseases was discovered, and they are known as infectious RNAs. My dear friends, today's my presentation is on infectious RNAs, the molecular parasites of crop plants. I will take another 45 minutes to tell what are these infectious RNAs, how they have been discovered, and why they are currently very, very important, and how they are causing different types of diseases in agricultural and horticulturally important crop plants. And these infectious RNAs were otherwise known as virides. I will come to that a little later. Okay, virides are nothing but very small, single-stranded, circular, low molecular weight, naked RNAs. Why I am using the word naked RNAs? There is no protein coat here, only RNA. Look at the sequences, okay? I told you the viral genome ranges from 1,000 to 250,000, okay, base pairs. Look at the sequences, nucleotide sequences. The virides are made up of just 250 to 450 bases, which able to cause diseases in plant. They are known as virides. Virides are nothing but very small, single-stranded, small naked RNAs ranges from 250 to 450 bases able to cause diseases in plant. They are called virides. Viride means, the literal meaning of the viride is virus-like particle, not virus. It is not exactly virus. Viride means it is a virus-like particle. It is nothing but naked plant-specific infectious RNAs. Naked plant-specific infectious RNA, tenfold smaller than smallest known viral RNA. They are very, very smaller. Okay, we will discuss little bit later how small they are. The virides are now regarded as the lowest step in the biological scale, and they are also considered as molecular parasites of the crop plants. We will discuss some of these things, very interesting aspects. So we know that I have already discussed the important characteristics of viruses. Viruses are nothing but nucleocapsid particles made up of nucleic acid, either DNA or RNA, externally surrounded by a protein coat. But here, look at this. They are very small circular RNA without a protein coat. That is the most interesting thing about virides. Viruses, they do code for protein. There are few genes which code for few proteins and enzymes. But in case of virides, even the genes are also absent, which lack the ability to code for proteins. In addition to that, another interesting thing about virides is that they have a catalytic property. That means ribosome activity. The small piece of RNA sometimes act like an enzyme and helps in multiplication of its own copies. So these are the important characteristic features by which you can distinguish virides from viruses. Virides from viruses. As you know that potato, it was in 1922, for the first time, there was a disease appeared in United States of America. It was better known as potato spindle tuber disease. Potato spindle tuber disease, where commercial potatoes crop, started showing a small spindle-shaped potato tubers. Okay, even commercial potato crop cultivation was affected by the appearance of this very unusual disease. Okay, so it has seriously affected commercial potato crop cultivation even in America. Though the disease was described in 1922, until 1967. Potato spindle tuber disease was universally believed. Even scientists believed that it could be due to viruses. They believed that the disease could be caused by viruses. It was only in 1971. T.O. Dainer, he was a scientist at the US Department of Agriculture, USDA, did several experiments and unable to isolate either the virus or the virus-associated nucleic acid or the protein coat. Therefore, his experiments to isolate the viruses associated with the potato spindle tuber disease failed. But on the other hand, he could be able to isolate a small piece of RNA which are infectious in nature and able to cause similar type of disease in healthy potato plants. Therefore, in 1971, he isolated a small piece of RNAs which are able to cause diseases and named these agents as virides. And he was the first person who identified the causative agent of potato spindle tuber disease, PSTD, as viride or virus-like RNA able to cause diseases. That was the starting point. To date, there are 32 different types of virides. More than 160 different types of variations of virides have been detected and are known to cause diseases in economically important and agriculturally important crop plants. And with lots of debate with T.O. Dainer in the International Congress of Virology, He was the Andre Loeb. He was the president of the Virology Congress. Finally, he, in his presidential remarks, he uttered the word, the time has come to accept virids. Till that time, we were dealing with only viruses. But in the International Congress, the Andre Loeb convinced that the existence of small infectious RNAs which are able to cause diseases in plants. This is, look at the picture. This is a just a symbolic representation of the PSTVD, potato spindle tuber viride, the size of the potato spindle tuber viride compared to other microorganisms that have been discovered from time to time. Potato spindle tuber viride, it is abbreviated as PSTVD. PSTV means virus, PSTV. This is how the international nomenclature. The viride is always named as VD, capital V, small d. Potato spindle tuber viride has been the first independently replicating subviral pathogen that has been identified. Look at the small picture showing potato size of the potato spindle tuber viride, okay, when compared to tDNA, the phage bacterial DNA, okay. It was the original preparation from the infected plant. Look at the genomes. I said the virides are very small. Viride genomes are extremely small and covalently circular because of similarity in certain sequences. When you isolate them, they become circularized. They look like a small circle. Sometimes the genomes adopt a rod-like secondary structure in vitro. Sometimes when you isolate them, these RNA molecules appear like a small rod. Some of the others, they looks like a hammerhead shape. Okay, the rod-like structures of viride contains 5 functional groups have been recognized. Virides are basically very small. I said their size ranges from 250 to 450 bases, and they are extremely small and covalent in nature. Because of the similarity in certain sequences, they acquire the shape of a small rods sometime, or they may sometimes looks like a small circles. So based on the configuration of these small virides, okay, rod-shaped configuration and hammer-shaped configuration, 2 types of virides have been recognized. One is the rod-shaped viride, the other one is Y-shaped or hammerhead-shaped viride, okay. So as I mentioned, there are 40 different types of virides have been identified in the world today, and all these virides can be classified into either rod-shaped virides or the hammerhead shape or the Y-shaped virides. Whether it is a rod-shaped virides or a Y-shaped virides, all these virides have major 5 structural and functional domains are there. The most important region in the viride is the central CCCR region, central conserved region, okay. Central conserved region in all the virides, the central conserved region is very common. On either side of the central conserved region, we have a pathogenicity region and we have a variability region. The pathogenicity region is believed to be involved in causing diseases in plant. The variability region, if at all, if there is a variability in terms of structure and sequences in 40 different virides, only at the variability region. On either side of the pathogenicity region and variability region, we have left terminal region and life terminal region. These are the 5 regions that have been identified: conserved, central conserved, pathogenicity domain, variable domain, left terminal repeat, and right terminal repeats. All these viruses, as I mentioned, they belong to 2 categories. So Paspiviridae, otherwise known as or rod-shaped virides, have been grouped under Paspiviridae. They are known to have a rod-shaped configuration. We have another type of viride, the quasi-rod-shaped or hammerhead or Y-shaped. They are grouped under another family, Avicen viridae. So these are the best examples, certain virides which have been characterized: PSTVD, potato spindle tuber viride; CCCVD, coconut kadang viride; ASSVD, Australian grapevine viride; HSVD, Hobstent viride; CBVD, Coleus bulimi viride. Okay, so these are all the important virides that have been characterized so far, which are known to cause severe diseases in both agriculturally and horticulturally important crop plant. I will not get into the details of all these viride, but these are the important viride. They are very, very important because they do cause economically important diseases in both agriculturally and horticulturally important crop plants. How do you detect them? So morphological methods very difficult because most of the symptoms are similar with the viruses. So very difficult to identify based on the symptomatology, based on the appearance of signs and symptoms and syndrome. Very difficult to differentiate whether the virus is causing the disease or viride is causing the disease. Therefore, earlier methods, even TO Daner started with spectrophotometric RNA analysis. With that help only, he could able to isolate a small fraction of RNA, okay, able to cause diseases. Later, powerful electron microscopes were invented, and then with the help of that, they could able to prepare a fine preparation of RNA. With that, they could able to prove that small RNA rods have been identified. As in viruses, bioassay on indicator plant still one of the best method to confirm the presence of association of viride with a particular disease. With the advent of molecular biology technique, slot blot hybridization methods, polyacrylamide gel electrophoresis have been used to confirm the presence of viride, small RNAs, and also differentiate from the host RNA. With the advent of molecular biology tools, we have RT-PCR methods have been successfully used, and powerful probes have been designed. With the help of these probes, primers, we can able to conduct a RT-PCR reaction, and we can able to detect the presence of all these virides in any crop plant which you are suspecting. So once you do the RT-PCR, subject the PCR product for cloning, sequencing, and phylogenetic analysis will reveal the association of a particular viride with a particular disease. As I mentioned, the bioassay on indicator plant is very, very much required. Once you isolate the small fraction of RNA from the infected plant, you need to conduct an assay, bioassay, on susceptibly important indicator plant. Tomato is one such indicator plant, and Cucumis sativa, cucumber, and Nicotiana benthamiana. These are the three indicator plant on which you can inoculate the sap and see within 30 days, you can able to see the presence of virides in them. And later you can do again PCR and you can confirm the association and multiplication of viride in the indicator plants. So Cucumis sativas, Lycopersicon esculatum, tomato, and Nicotiana benthamiana, these are the three indicator plant that can be used for conducting bioassay for virides. Unlike viruses, virides do not code for any proteins. So how do they replicate? Okay, how these viride infectious RNAs replicate? Viride's replication mechanism uses RNA polymerase II enzyme, the host cell enzyme. So all these virides use the RNA polymerase II enzymes of the host for replication process. Polymerase II synthesizes new RNA using the viride's RNA as template. Okay, some virides, as I mentioned, the hammerhead type of Avicen viridae, the members belong to Avicen viridae, they have a ribozyme activity where they have a catalytic property, they replicate themselves, which self-cleavage and ligation do takes place. Look at this particular picture. It has been proposed for the multiplication of the virides. So the moment the small circular RNA, which we consider as a plus strand, which is able to cause diseases in plant, a small RNA when enters into the plant system, the plus strand utilizes the nucleus encoded polymerase II enzyme, and on which a new strand is synthesized, a new RNA molecule is synthesized. Okay, so because of the ribozyme activity, the self-cleavage activity, it gets linearized on the RNA ligase, joins this linearized molecule into a minus strand circular form. The circular form of viride acts like a template on which plus strand is synthesized, plus strand of RNA is synthesized. Again, because of this ribozyme activity, it gets undergoes self-cleavage and it forms a linear RNA molecule. Again, the DNA ligase enzyme which helps to ligate the linear molecule into a plus strand viride. The plus strand molecule known to travel inside the phloem sieve tubes, and it is systemically transported to other parts of the plant body through plasmodesmata connection. It is the same picture I have already explained. Though these infectious RNAs, they are so small, little is known about the mechanism by which virides cause such a severe diseases in crop plants. And in the severe, infected plant, it has been observed that cell wall malformation, membranous bodies with distorted organization, complete dislocation of the plasma membrane, chlorotic disturbances in the cellular level, changes in the expression of pathogenesis related proteins, alteration in the hormones, the flowering hormones gets affected, and certain metabolites get also affected at the biochemical level. All these have been very well analyzed, but the exact mechanism by which they induce diseases is still yet to be unraveled. Some virides cause no detectable symptoms, even though they are present inside the host plant. There will not be detectable symptoms, okay? But there are certain virides, when they enter into the plant system, they do cause severe damage. And it has been studied that how do they interfere with the host metabolism or host functional genes, and they have suggested a gene silencing. The pathogenicity genes which are residing on the viride molecule, they are known to suppress the functional expression of the genes through the gene silencing mechanism. Therefore, the functional mRNAs gets degraded. As a result, different types of symptoms do appear in the infected plants. These virides are known to travel from one part of the plant body to another part of the plant body through plasmodesmata connections and through the phloem sieve tubes. And there have been a lot of experiments have been conducted to prove that they go through plasmodesmata connections. Many host plants are known to harbor different types of virides, which include both monocotyledon plant and also dicotyledonous plants. Many ornamental plants, vegetable crop plants have been affected by different species of phosphorvirides and also avicen virides. And therefore, there is a systematic study has been conducted, and the studies have revealed that there are 40 different types of virides have been isolated, and they have been very well characterized. Based on the detection methods, powerful detection methods that are available from different parts of the world, virides have been detected, and they are known to affect severely, cause different types of diseases. How do these virides spread? The virides are known to spread from plant to plant through vegetative propagation units that we use, propagative materials that we use, maybe tubers, cuttings, or bulbils. When we use the vegetatively propagated material, the vegetative propagating units, they do contain the virides and responsible for appearance of symptoms in the later days. Mechanical transmission and agricultural implements. Several agricultural implements that we use in the agriculture field, they do spread the virides. Infected seeds and pollens, they do carry the virides, and therefore they are considered as known to transmit the virides. Phloem sucking aphids, certain insects, especially phloem sucking aphids, are known to transmit the viride from infected plant to uninfected plant. This I have already discussed. Then how to manage some of these diseases? At present, there is no currently available management practices, particularly the diseases caused by virides. Using only viride-free nuts and seeds is the only method to reduce the appearance of viride diseases. use of resistant varieties, eradication of infected plants, insect control to reduce the disease incident, and following the phytosanitation method. These are the important methods that are available currently for the management of the disease. I would like to give few examples how viroids are very important to us and how they are affecting our agriculturally and horticulturally important crop plant. Look at this particular picture. It is a coconut kadang-kadang disease occurred in Philippines. This is a coconut kadang-kadang disease. It is a disease of coconut, Cocos nucifera. Okay, this disease was first appeared in the Philippines in 1927 and 1928. By 1962, all the 25 million coconut palms died due to disease. So far, there are 40 million coconut palms have been killed since the first appearance of the disease, and the economy of the Philippines is now completely jeopardized. They are not going coconut, and it is not a livelihood crop for people of Philippines now. And when the disease comes, there are 5 different stages have been recognized, and by the time it reaches the 5th stage, the entire coconut gardens gets destroyed. So in different, but not only here, in the other regions of the world also, many coconut gardens have already been destroyed. Since 1927-28, very recently, the causative agent of the coconut kadang-kadang disease was identified. The RNA, infectious RNA was isolated and genome was sequenced only in 1981. And the genome sequence revealed that the RNA is made up of just 246 nucleotide molecule, and it has central conserved region and other 4 important region, very important, which is normally present in all viroid. CCCVD is the number of different molecular forms, and it is also known to cause diseases in other regions of wherever coconut is grown. This is the original picture showing purified coconut kadang-kadang viroid RNA preparation. You can see the presence of small circular molecule. They are nothing but the small RNA molecules isolated from coconut kadang-kadang disease affected Cocos nucifera plant. Later, Randalls, they also identified coconut Tinangaza viroid. It is a variation of the coconut kadang-kadang viroid, which is also known to cause similar type of disease in Tinangaza region. I have already told you the potato spindle tuber disease was the first viroid to be identified, which was associated with potato tubers, spindle tuber diseases in potato. And look at the symptoms. Since the first discovery in 1971 by T.O. Daner, different variations in their pathogenicity to cause diseases have been isolated. Different types of viroids have been isolated. Though they are PSTVD, lot of variations, strains have been isolated, and they are known to cause severe diseases in potato crop in different parts of the world. The same potato spindle tuber viroid is also known to cause a potato, similar disease in tomato, because both potato and tomato, they belongs to the same family of Solanaceae, and now it has been revealed that potato spindle tuber viroid is known to cause disease in tomato plant. Tomato chlorotic mottle viroid, TCDVD, it is one of the severe disease, has been reported in different parts of the world. It also been recently reported from our lab from India. And the viroid also known to cause disease in pepper chart fruit. Okay, we have bell pepper, very important vegetable crop. Okay, now they are known to cause diseases in bell peppers. When there is an infection, the size of the bell pepper is reduced to small size, and the marketable quality of the fruit size has been reduced. Very recently, the Indian apple scar skin viroid has also been reported, and which directly affect the appearance, texture of the apple quality. Okay, look at the apple skin scar viroid, which is completely going to damage the appearance of the apple, and therefore it completely affects the marketable value of the food, fruit. Therefore, it is very important virus, and it has been reported from different parts of the world, and Indian apple scar skin virus also been reported from Himachal Pradesh. Apple dimple fruit virus, this is the variation of the another virus, and it is also known to cause similar serious diseases in an apple. Another virus, which is known to affect the grapes, has been recently isolated, and these are the typical symptoms of the leaves of grape plants, grapevine, showing typical symptoms of virus. Citrus exocartis virus, CEVD, it has been reported from India also, and we know that Indian citrus fruits known for the better quality fruits, but nowadays these citrus exocartis virus severely affected the production of the citrus fruits. And one of the important characteristic feature of citrus exocartis virus is that prominent bark cracking and splitting, by which we can identify the disease symptom which is caused by citrus exocartis virus. Citrus bent leaf virus, this is another virus which has been reported in citrus plant. Chrysanthemum chlorotic dwarf virus has been reported. You know that chrysanthemum is an important floricultural crop in different regions of the world. Also in India, it has been reported from India. Chrysanthemum stunt virus has been reported. Whenever there is infection, the small plants fail to flower in the blooming season, and therefore the yield, the flower production and yield will be completely affected. Columina latent virus has been reported very recently, and it is known to cause severe diseases in tomato. And avocado sun blotch disease, it is caused by avocado sun blotch virus. Look at the fruit size, quality of the avocado gets affected. Peach latent mosaic virus has been reported in peach plants, and cucumber mosaic virus, small cucumber mosaic virus has been reported, and it is known to affect the quality and size of the fruit side. Coleus bloomy virus has been reported from one of the important ornamental plant. Even from India, it has also been reported, which is known to affect the quality of the flowers. You know that obstant virus, HSVD, okay. The obstant is humulus lupulus. Particularly, the female strobilus of these plants are used in beer production. And these female strobili, which contains so many betel ions, and they impart characteristic flavor and stability to the beer. Because of obstant virus, the obstant hops cultivation in Slovenia completely affected, and now we are producing a beer without the humulus lupulus female strobilus. This obstant virus is known to affect the mulberry leaf quality, and very recently it has been reported that the obstant virus is known to cause similar type of diseases in mulberry. In mulberry, the leaf quality is very, very important. Whenever there is an infection, the leaf quality gets affected. So also the cocoon production gets affected severely, and therefore it is very important disease. And in India also, it has been recently identified. So these are the important symptoms which are I'm just restricted myself for a few important economically important cropland and disease caused by the virus. There are so many other things to discuss. Because of lack of time, I am myself restricting myself to few important things. Is there any virus infecting humans? That is the question. It has been reported, as I discussed earlier, the RNA is the material infective agent, a smaller RNA, which is able to cause diseases in plant, which we consider as virids. Is there a virid like RNAs able to cause diseases in plant and animals? Hepatitis delta virus, it is abbreviated as HDV, which infects humans, has many viride-like properties and may be related to virides. It has been reported. HDV has a circular RNA genome, hence HDV is often called as virides of animals. But interestingly, the HDV requires a helper virus, one more helper virus to multiplication. In that context, the HDV virus differ from the viride which cause diseases in crop plants. The origin of virides. So you may be like how these the viride, small infectious agent, originated. The question, there are different theories have been proposed from time to time, and the people believed that the virides are nothing but primitive viruses without any biosynthetic machinery. With small genetic information, they are still surviving in wild plants. That's what some people believe that. But others believed that they are the degenerated viruses. They developed from low molecular weight RNAs in the first, and they degenerated, and they are still surviving in crop plants. Others believed that they consider them as abnormal RNAs, nothing but the nuclear RNAs from eukaryotic cells. They must have escaped from the cells, and they become pathogenic by mutation. This is another theory. They believe that they are nothing but abnormal RNAs, and they are able to cause diseases. In spite of these theories proposed from time to time, the T.O. Dainer, the discoverer of the viride, in 1989 hypothesized that virides may represent living relics from a hypothetical ancient and non-cellular RNA world of DNA or protein. So if you go back to the origin of life, we all believed that the protein, chemical molecules originated first from DNA, RNA originated from RNA, the different types of protein. That's what we discussed in the central dogma. But the T.O. Dainer believed that much before the evolution of RNA, there was an RNA world. From RNA world, these, the virides are nothing but the living relics. Okay, they are still living as causative agents in different types of plants. They are nothing but the ancient non-cellular RNA world. With these, few points, which I would like to conclude by saying that virides are very curious subviral infectious agents, smaller than viruses, which are known to cause diseases only in plant. So that's why they are very interesting. They come under emerging plant diseases. Many are causing diseases which are considered emerging plant diseases, and many wild plant, whether they do harbor such viride, we do not know. So the time has to reveal that whether wild plant reservoirs of viride, we need to investigate. The another interesting thing is that the minimal genetic information. The RNA, nucleotide sequences is so minimal, 250 to 450 base pair, made them very curious agents. Okay, but though they are very small, they are able to cause very severe diseases in agriculturally and horticulturally important crop plant. Identification of the diseases caused by viride is a challenging one, and we need a powerful tools are required for diagnosis detection of these virides. So more studies are also required to address the issues related to appearance of symptom, mode of spread of disease causing agent, and agents which are responsible for transmission of these viride diseases. Best way is to manage diseases by using phytosanitary measures and also seed fortification program. So, use of disease-free seeds, viride-free seeds can be used. That is the only method. But with the advent of molecular biology and genetic engineering, people are also trying to use the biotechnological approaches, including transgenic and gene silencing methods to silence the virides, the virides, and thereby they want to reduce the effects of viride, which are able to cause diseases in plants. With these remarks, I would say that The Louis Pasteur once upon a time stated that the importance of the infinitely small is infinitely large. That is true with the small RNAs, which are causing diseases in plants at the moment. So with this remark, I stop my presentation at this particular point. If there are any specific questions, I will be very happy to answer. Hello.
Hello. Any
Hello,
questions?
yes.
Participants, teachers, any questions?
Sir, yes, sir. Sir, can I?
Yeah, please.
Sir, very good afternoon, sir. Thank you, sir. And I do have a couple of questions, sir.
Yeah, please tell me.
Number one is that how these viruses are said to be stable, because these are RNA molecules. You said about their RNA viruses, and they are like RNA. You know that RNA are really unstable at any conditions, but these are said to be very stable. And that is my first question. And my second question is that you said they are transferred from plants to plants, and how they are able to move, or how they get transferred. How they are moved from plants to plants, or how they are get into the environment. That are my two questions.
Yeah. Isolation of small RNA is very difficult. Okay, and most of the time they get degraded by RNAs. Okay. So, but lithium chloride extraction methods have been standardized. Okay, and using lithium chloride extraction method, you can isolate them. Okay, and immediately you can do the PCR. Okay, and thereby you can detect the presence of viruses. That is one thing. Okay, regarding transmission of viruses, only insects are known to transmit. The phloem sucking insects are known to spread the virus from infected plant to healthy plants at the moment. There is no other, but people also detected the presence of virus in pollen grains and also some seeds, okay, from the infected seeds. If the mother plant is infected, the seeds collected from the plant is likely to have virus in the embryonal region, okay. So therefore, there is a very possibility that the virus infectious RNA is likely to get transmitted to next generation.
Okay. Thank you. Thank you.
Yeah.
Sir, good afternoon, sir.
Yes.
These viruses doesn't have any code protein or doesn't have a definite structures. Like when host RNA polymerase 2 replicates this mRNA and it moves from one cell to the another cell, right?
Yes.
At that time, why don't host nucleases doesn't recognize this RNAs? Like, is there any special kind of modification it undergoes protection from the host nucleases?
Maybe that could be one of the reason. Okay, so they do sustain the effects of RNAs, host RNAs. Okay. Yeah.
Like, did they identified any specific? For example, our host DNA undergoes methylation basically in order to protect from our own nucleases, but this RNA undergoes any such kind of modification in order to protect from the host RNAs. They have identified it?
No, sir, no such work. It is still going on. Lot of work is going on as far as molecular biology of virus is considered. It is one of the hot topic for present day plant pathologist and molecular biologist. They are trying to understand the molecular mechanism by which they sustain the effects of the host host effects.
Yeah, because it doesn't have any code protein, it doesn't have any protection layer, like unlike normal viruses. It's very as you said, it's still a mysterious then.
Yes.
Maybe they have some special mechanism to resist this degradation.
So
that's what, whether they form like duplex structures or something, three-dimensional structures in order to protect from nucleus. We don't know. Okay, thank you, sir.
Yeah.
Sir, very good morning, sir. This is Kaliswaram from Tamil Nadu.
Please tell me, sir.
Sir, I have two questions. One is common question.
Yeah, please tell me.
So, beginning of your slide, you told about that agriculture field, one of the growing field today. But fortunately, I saw many newspapers, cuttings, everything. So tell about that. Peoples are in the village, including village peoples, are moving towards the urban area. They are leaving their agriculture land. They are selling for plot purposes or many other purposes.
Yes.
How this statement can be justified? This India is glorying in the field of agriculture.
No, because India agriculture is the backbone of Indian economy, and 70% of the population were dependent on agriculture. Okay, earlier, maybe 1970s and 80s. Today, if you look at the statistics, the Ministry of Labor and other department, only the 42% of the Indian population is involved in agriculture. So that is why the number, the percentage reduced from 70% to 70% to 42%. Okay. And as you have mentioned, it is true that the rural, young rural Indians are migrating towards cities. Yes, it is happening. And at the same time, the engineering graduates, after obtaining their degree, they're coming back to the villages and they started doing agriculture. Is what we call smart agriculture. Okay, it is not the traditional agriculture. What they are doing, the smart agriculture. Okay, so in thereby the agricultural activity has picked up again. But again, unless even in the rural areas, even our farmers have been given the modern agricultural equipment and know-how technologies of modern agriculture, and they all adopted modern agriculture methods, and they are increasing their agricultural output, and they are stabilizing their economy, and all these things are contributing a lot for the growth of Indian agriculture.
Okay, sir. Sir, next question, sir.
Yeah.
The insects are transmit the disease, the viroid, to the next plant. So what happened inside the insects? Is there any evidence? Is there, sir? Or
yes,
infection on insect?
There are different kinds of insects. They do harbor the viruses. Just like viruses, they do harbor viroids. When the insect, there are certain phloem sucking insects. When they suck the juice of the sap from the plant, they do acquire the virus, viroids, and also bacteria. For certain period, these viruses, they do survive in the midgut of salivary glands of insects. Before they get killed, they need to go and feed on other healthy plant, so that every possibility that the viroid may gets transmitted to other healthy plant.
My question is, sir, whether the viroid infect the insects also. Is there any evidence, sir?
There is no such in insects have been phytoplasmas have been detected in insects. Phytoplasmas are again wall-less bacteria known to cause diseases in plants, and certain insects are known to acquire some of these phytoplasma-like bodies in their salivary glands. Okay, but there are no such viroids have been known to survive in the insect midgut, but they have been detected in the mouth parts of certain insects.
Okay, sir. Okay, sir. Okay, sir. Thank you, sir.
Yeah, thank you.
Thank you.
Any questions? Okay, if there are no more questions, so someone can propose vote of thanks.
Okay,
please.
Thank you, sir. It's really honor to me to propose vote of thanks to Professor G.R. Janardhan, sir. Thank you, sir, for your great time and highly informative lecture. My heartfelt thanks to you, sir. Thanks again.
Thank you very much. Thank you very much.
Thank you, sir.
Thank you, sir.
I thank the Academic Staff College of University of Mysore for giving this opportunity to share my presentation. We have done some research, but I could not share those things. Okay, if you are because of time factor, we are the first to report the association of viroid in India. We are also working on grape viroid and also on mulberry problem in mulberry. Okay, So I had a DST sponsored research project. With that, we could able to publish, identify 2 different types of viroids associated with tomato and also mulberry. Yeah, thank you very much.
Thank you, sir. Thank you, sir. My side.
Thank you very much. Thank you so much.
Thank you, sir.